Okay, so I think it is 4 o'clock now. We're going to get started here right away. So we're going to pretty much pick up where we left off last time. Before we get there, I just wanted to talk a little bit about the lab. This afternoon, I sent you a little bit of information with the lab. It's more of an introduction type of guide. It's about maybe 3 pages that talks about the lab and what we're going to be doing, evaluation. And then there's an appendix along with that that includes lots of detailed instructions on the lab reports. And so I'll be talking about that a little bit more next week when we have our first lab. I will have that available by Thursday at the latest. I might be able to get it up earlier than Thursday, so I will discuss this next week, probably talk about it on Wednesday. I think there was something else I was going to say about the lab. Just on this slide here, if you've not done so, complete the plagiarism course and sign the certificate, and you can upload that to Moodle. Just scroll down where it says laboratory, and there should be a spot there to upload your certificate. And in the meantime as well, please take a look at the lab guide, and we will be going through different parts of it as we start to do the assignments. The last thing I was going to say is feel free to try the chat function if you have any questions. I may not see it right away, but at least that way, if you have any questions, I can respond and do that in real time during the lectures. Okay, so let's get into this. Last time we left off, we were talking a little bit about a history of cell biology, and there were just a couple of things to kind of finish up on. This really brings us to the 1800s, so this is really quite incredible. This is a lot of this stuff happened 150 years ago, and this is the cell theory. This was developed by Schleiden and Swan. So these two guys, I can't remember which was which. I think Schleiden was a botanist and Swan was a zoologist, and they were getting together and they were talking about their findings, and they had realized that pretty much all the samples they looked at, that their samples were made from cells. And so they came up with something now that we all call the cell theory. So this is the idea that living things are made from cells, that the cell is the basic unit of structure and organization in organisms, and that all cells come from pre-existing cells. People have added some modern kind of twists on this in terms of just kind of adding to cell theory, the whole idea that cells are dealing with energy metabolism, and that hereditary material is an important part of cells. I see there's a question here. Somebody is saying, do we need to do WHMIS for the lab? And the answer in that is no. Since we're not doing any labs in person, you don't need to do the safety course. You can still do the safety course if you like. You might learn something. If we are doing in-person labs next semester, you'll need to do it then anyway. But don't do it if you don't need it for any courses. There are, of course, many types of cells, just even within your own body. You've got red blood cells, you've got neurons, you've got skin cells, you've got liver cells, all sorts of things. And of course, there are many different types of organisms. And so we'll be touching a little bit on this here or there. Mostly we'll be focusing on kind of things that are common among cells. So one thing that is going to be a bit of a theme in this course is talking about what is a eukaryotic cell and what is a prokaryotic cell and what the differences are. And so you may not know this, but it's actually right in the name. Eukaryotic, I think I can write on my screen here with this pen. So this is Greek, by the way. And eu means true. And karyote means nucleus. So it's right in the name, true nucleus. You can probably see that from the diagram. There's 2 things you notice right away from this diagram. The differences between the cell types. Obviously, the eukaryotic cell is bigger and it has a nucleus. Those are 2 of the main things. Prokaryotic, pro, by the way, pro means before in Greek. And so whoever named eukaryotes and prokaryotes, we're thinking that prokaryotes were more primitive or they existed before eukaryotic cells. So we are going to, this is going to be a theme all semester talking about this goes on in eukaryotes, this goes on in prokaryotes, and we're going to compare them in quite a bit of detail at some point, probably starting next week. Here's a typical prokaryotic cell. It's really hard to say typical. There really is a huge amount of variety in these things. The main thing to say is they have no nucleus, no membrane-bound organelles, but they do have quite a number of features. You can see they've got a cytoplasm, ribosomes, they have a cell membrane, all the usual stuff. So prokaryotic cells involve two kingdoms. We have the bacteria and the archaea, and I will come back to them in a moment. Here's a typical animal cell. This is a eukaryotic cell. You can see it's a lot more complex. You've got all those organelles that you learned about in high school. We've got mitochondria, Golgi body, endoplasmic reticulum, and so on. And so again, much more on these as we go through the next few units. So we can classify cells. This is what I want to talk about now, about classification of organisms. We can classify cells by cell type. We have the prokaryotes, which includes 2 domains, and we have the eukaryotes, which includes 1 domain and many kingdoms. You're probably familiar with animals and plants and fungi. Sometimes we talk about the protists, and sometimes we talk about the algae. Sometimes people group them together. Protist is really kind of one of those weird catch-all names that kind of just means it's a eukaryote and it's not an animal, plant, or a fungi. Often it's unicellular. But other than that, there's a lot that could or could not be said about protists. Like I said, usually unicellular, but there are many examples of protists that are not. Just keep that in mind. You know, this is actually kind of a general kind of rule about biology, is that pretty much every sentence in biology we can put the word usually in front of it. Usually eukaryotic cells have a nucleus. But I can think of an example that don't. Your red blood cells, once they're mature, they don't have a nucleus anymore. Usually bacteria do not have other organelles, but there are a few weird bacteria out there that do have organelle-like structures. So you can use that word usually many times. Later on at the end of the course, we're going to talk a little bit about viruses and prions. These things are not cells. They're not usually considered living. Much more on them later. So here's just another way to look at the tree of life. You can see we have our bacteria over here, and we're going to talk a lot about these. And just looking at that particular chart, I don't see E. coli on it, but we're going to talk a lot about E. coli. We also have our archaea over there. Again, we're going to be looking at this and talking about them in a lot more detail. And those, of course, are our prokaryotes. And then we have our eukaryotes, and you can see there's our animals, our fungi, and our plants. You can see that fungi and animals are actually a lot more related than fungi and plants. We have similar methods of feeding. We both eat things, whereas plants make their own food using photosynthesis. So you can see there's a note here. It says these are based on DNA sequences when people do these charts and try to show relatedness of things. So we'll talk about that a little bit more in a few minutes. So there's our bacteria. Bacteria are prokaryotes, no nucleus. They have a certain type of phospholipid. They're unbranched. And other than that, like I said, we're going to talk about them in a lot of detail. There's our archaea. On the surface, they look a lot like bacteria, similar size, no nucleus, but when you start to get down to the nitty-gritty details and look at the biochemistry of these things, they have branched phospholipids, they have completely different RNA polymerase enzymes. There's all sorts of weird things that they can do that makes them quite a bit different from both the bacteria and the eukaryotes. And there's our eukaryotes. You can see we've got the animals, plants, and fungi, and protists. Much more on these, like I said, in the next few weeks. Okay, so what I want to talk about are 2 things. One is taxonomy, and just kind of give you an example of how taxonomy works. I'm going to show you how we might classify a human. Now you're probably wondering, do I need to learn all this? Here's what I care about. I care about domains, care about genus and species, and pretty much everything else in the middle. Not so much. So let's start looking at what a human is. So for domains, we had three options, right? We had the eukaryotes, we had the bacteria, and the archaea. So our cells have a nucleus, so that makes us eukaryotes. We're animals. So what is an animal? We are motile, we are heterotrophs. That means we feed on things. We're multicellular. And so those are a few things that make us animals and not, let's say, fungi or plants. What phylum are we? We have a nerve cord, so we're chordata. Class, mammals. Mammals have hair, and we usually feed our young milk. So that's where the word mammal comes in, for mammary gland. We're also primates. So that means we have opposable thumbs for grabbing things. Most animals do not have anything like that. There is one other order, I can't remember the name off the top of my head, of organisms that have opposable thumbs. I don't know if anybody knows what it is. Sometimes you go to the zoo and maybe you see these things, pandas and raccoons. What family are we in? We're hominids, which means we have a large brain case and no tail. And then what we care about are genus and species, Homo, and you probably know that we are sapiens. What does sapiens stand for? Well, the nice thing is if you get to name yourself, you get to call yourself something nice. So we call ourselves wise or sentient, which kind of means thinking. You know, that's something that, you know, seems to distinguish, at least for the most part, distinguishes us from animals. Not that animals don't think, but certainly humans are a little bit more advanced in that category as far as we know. So I want to show you another example of a mammal. Here's another mammal. So this is a dog, and you can see the first few levels of classification are very, very similar, right? So, you know, they're a eukaryote, they're an animal, they're a mammal. After that, though, there are differences between dogs and humans. So dogs are carnivores, so they eat mostly meat. They have special teeth. That's something that's looked at. If you look at our teeth, we're definitely omnivores, not carnivores. What's family? Canines are dog-like things, so that includes foxes and coyotes. It does not include hyenas, apparently. I don't know what group hyenas are in, but they are not dog-like, apparently. And then the genus is Canis, and the species is Lupus. So Lupus means wolf, right? So you probably know that dogs and wolves are the same species. They can interbreed. Dogs are really, you know, basically domesticated wolves. So here's a fun fact for you. Take a look at this word here, lupus, right? So some of you may have heard of this little book series or movie series called Harry Potter. And remember the third book, The Prisoner of Azkaban, and there was, what was his name? Professor Lupin. Professor Lupin. And you may remember the secret at the end of the book that Professor Lupin was a werewolf. Well, you can see right in the name. The name was inspired by the Latin word for wolf. So the author was giving everyone a little bit of a hint as she went along as to what was going on. So one more thing about dogs is sometimes we also talk about subspecies or breeds or strains. So it means it's the same species, a dog and a wolf can breed together, but sometimes there's enough of a difference that we may actually talk about different breeds or subspecies. So the domestic dog is actually called the Canis lupus familiaris. Familiaris means friendly or domesticated or something like that. Okay, so I want to show you an entirely different type of example. The third example is a microorganism. So this is a bacterium called Staphylococcus epidermidis. So take a look at this. Right at the very beginning, it's an entirely different domain. So it's a prokaryote, it's a bacteria, which means it has no nucleus, and it has certain biochemical things like unbranched phospholipids. We look at the kingdom, it has a certain type of cell wall that we call gram-positive. We'll talk about that in a couple of weeks here, what exactly that means. Firmicutes. So now, you know, sometimes when you look at bacteria, you can see the shape of these bacteria, right? They're kind of just round. Well, it turns out there's dozens, hundreds, probably thousands of species that are kind of round. So how do we start looking at the characteristics of these things? We have to sometimes start looking at the DNA and see what's going on. In this case, firmicutes, for some reason, have less G's and C's than A's and T's. So something like 30%. Bacillus, based on 16S RNA. Ciliales, based on 16S RNA genes. And now we can actually look at something that's a little bit more to do with the characteristics. So Staphylococci. So staph, you can see it says here means cluster, and coccus means sphere. So these cells are spherical shaped, and they're clustered together, and they kind of look like a bunch of grapes. So now we have a name that is based on a physical description of it. That's also the genus name, basically, Staphylococcus, and the species name is Epidermidis, which is because it lives on the skin. So we have a descriptor here. So let's talk about what is a species. A species, you can see I have a definition there. Classically, biologists will define a species as something, organisms that can interbreed with one another. So the example of the dog and wolf are a great example of a species, right? They can breed, they can produce fertile offspring. I'll show you a couple examples here. I can advance this slide. There we go. So there's two species there. We have donkeys and we have horses. So if you get a male donkey and a female donkey, you get baby donkeys, and they're a species. Same thing with horses. You get a stallion, you get a mare, and you put them together, and you get baby horses. But if you put a donkey and a horse together, and well, farmers have been doing this for a long time, you get something else which is called a mule. So it turns out a mule is not a species. You can't take two mules and breed them. So it is a living organism, and it's a useful organism for carrying packs and things like that, apparently. But in itself, it cannot breed, so it is not considered an actual species. I'll show you another one. This one's just for fun. So what happens if you get a zebra and a horse? I know you're all thinking, okay, is it a hebra? Is it a zorse? Well, it's a zorse, of course. So I hope you like my joke. So this is also a sterile hybrid, not an actual species. Okay, so where am I going with this? Well, it turns out that when we start to look at microorganisms such as bacteria, and this gets way more complicated. There's no male and female E. coli, for example. It just doesn't work that way. There's no genders. They don't often even mate. They just reproduce asexually. Sometimes they will actually exchange DNA, and they will do that, with actually unrelated species. So what is going on here? It makes it a lot harder to define these things if we're defining things based on how they can actually breed. So what do we do for microorganisms? And actually much of the biological, the organisms found in the biosphere is we can look at taxonomy, which is looking at characteristics. So characteristics in a dog, has hair and canine teeth, etc. In microbes, we can look at other characteristics like cell shape. Does it stain purple or pink when you do a Gram stain? The biochemistry, what kind of phospholipids it has. And we can only get so far with that. So thankfully nowadays we have many genome sequences, and we can use phylogeny. So phylogeny, you can see taxonomy, has to do with physical characteristics. Physical characteristics. Sorry about that, I'm just learning how to write with this pen. And phylogeny is relatedness based on DNA sequences. Usually we look at the rRNA. So what is the rRNA? That is the ribosomal RNA. So what's the big deal with the ribosomal RNA? Well, if you think about it, if I were to look at human genes, let's say the gene for hair color or the gene for hemoglobin, that's great if you're comparing mammals, but plants don't have hair, plants don't have hemoglobin, never mind bacteria. So what we're doing is trying to look at something that all organisms have, and all organisms have ribosomes. So it's a good frame of reference. That's why we use ribosomal RNA for phylogeny. So by the way, I'm not doing a lot of notes this week, but starting next week, usually what I do is I write a lot of notes on the whiteboard, and starting next week, I'm going to be starting to write a lot more notes. I'll probably use Word or something like that, and I'm not going to provide you with those notes. You're going to have to write them down yourself as you do the lecture. And why am I doing this? I want you guys to work hard. I want you to pay attention. I want to give you value to your lectures. And a lot of these notes will either be helpful with your studying or they'll be helpful for preparing for midterms. So pay attention to what I'm writing down because these things are important. For example, right here, taxonomy, phylogeny, right? That would be a good midterm question. So how does phylogeny work? Well, kind of like this, right? Nowadays we have computers. It's very easy to do. You get the sequence of one organism, or at least the gene for ribosomal RNA, and you get the sequence for organism number 2, and you can compare. And you can see in this case here, you know, I'm comparing 1, 2, 3, 4. Those nucleotides are the same. The next one is different, and you can get a percentage. Are they 99% the same? Are they 98%? Are they 63% different? That kind of thing. Here's another example. You can see that these two organisms are a little bit more distantly related. And so we can compare things and see what, whether we can find, you know, are the organisms the same or are they different? So how do we know if we have a different organism? Kind of depends on who you're talking to. But there is kind of a bar for at least microorganisms, about 2 to 3% difference, and then you have a different species. So if two organisms are 99% the same, they're the same. They are the same organism. If they're 98% the same, they're the same organism. 97, now you're getting in that gray zone. 95%, now they're probably related, but not the same organisms. Humans, by the way, males are 99.9% the same with one another, and females are 99.9% the same with one another. But males have that Y chromosome, so if you look at males and females, we're still in the 98 point something range anyway, in terms of our genetic content. So I showed you this, this nice little, I guess, tree of life, or whatever you want to call it, phylogenetic relationships. And this is actually from an older version of the text. The newer version, I didn't like the picture quite as much. I also found this one here. You can see this is something that certain biologists are doing all the time. They're trying to, you know, see what these things are, how they're related, and, you know, this is something that scientists like to do. We like to classify things and whatnot. I want to show you this diagram because this person is saying that, hey, look at this. According to their analysis, eukaryotes and archaea are actually much closer together in terms of relatedness than the bacteria. Anyway, interesting article. If you want to check it out, the link is there. You can look it up. Okay, so something important for your lab reports and all of your assignments is to know all about the binomial naming system. Okay, so you may have heard of this system here. Sometimes we call these things the Latin names of organisms or the scientific names. Binomial, of course, bi means two, and nomial means name. So it means the two naming system. This was proposed way back in the 1700s. Why do we use Latin and sometimes other dead languages? Because we're trying not to show any favoritism, right? You know, favor English over French or any other language kind of thing, right? So that's what was chosen. Obviously, it was a European who developed it. The names are going to be italicized or underlined. This is really important. If you do not italicize or underline species names in your assignments, you will lose marks. So do not forget. Every organism has two names, genus and species. So the example of Homo sapiens, there's the genus, there's the species. Notice it's italicized. Notice the genus is capitalized and the species is lowercase. And so often in your reports, you might write out the full name the first time, and then the second time around, you might just abbreviate the genus by giving the letter. So you might have H. sapiens, or you might have E. coli, and so on. So let me show you some names of some organisms and how they're usually named. I have a whole bunch of examples here. So all the best names are descriptive. So we saw the descriptive name for a dog or a wolf. A cat, in case you're wondering, is the Felis domesticus, so it means domestic cat. We talked about Staphylococcus epidermidis. So here's another Staphylococcus. Remember, staph means cluster, and coccus means spheres. So we have clustered spheres like that. Okay, And notice the second name here, aureus. So if you're taking chemistry, or if you have in the past, you may remember that particular element there. Everyone seems to remember that one because it means gold. So let's take a look at these cells. I have some pictures. So take a look. Those cells are clustered grapes, just like the Staphylococcus epidermidis. Unfortunately, the name epidermidis was already taken, and this is another skin organism, so we name it after a different characteristic, the color it looks like on a petri dish. So you can see it's a nice goldy yellow color, and so the name is relatively descriptive. Good way to name things. Another example. Here's another interesting example. I found this example in the Guinness Book of World Records. So take a look. We have another coccus, right? So coccus means sphere. Dino means terrible. It's the same dino that you see in dinosaurs. I don't know why the spelling is different, but dinosaurs means terrible lizard. In this case, we have a terrible coccus or terrible sphere. And then radiodurans. It means resistance to radioactivity. So you're thinking, what now? That's right. This particular organism was found in a nuclear reactor. You know, one of those places where there's probably, you know, hundreds of times more radiation than a human could survive, and they find this bacteria. They've been studying it, can survive all sorts of things, cold and dehydration, acids, and so ended up in the Guinness Book of World Records. Good name for it though, Dinococcus, I like that. Sometimes, if you're lucky or unlucky, you might get something named after you. So Escherichia, Theodore Escherich, was a pediatrician who was studying bacteria in infants, and when he died, they named it after him. So I don't know if I would be too happy if somebody were to name fecal bacterium after me, but that was the case, and he's dead and gone, so he doesn't have a choice. Coli is for colon because it's found in your intestine. Other names here, Listeria. There was a man named Joseph Lister that was named. Listeria was named for. How about that one? Maybe some of you guys are Rolling Stones fans, and whoever named this organism was a fan of Mick Jagger from the Rolling Stones, so that's why this organism was named that way. How about this one here? Anyone else a fan? Take a look. So I don't know if you are a fan or not, it doesn't really matter, but I will show you. This is a wasp, and the wasp was named after Donald Trump. Whoever saw the wasp, they could not help but be inspired by the yellow comb over. And they're like, the name has to stick. So there you go. It was named in honor of him, or at least his looks. So hope you enjoy that one. So scientist names, sometimes you end up with a few other weird things out there. I found this one here named after a geographical location. So you can see here, Long Beach, California. Thought that was kind of cute. And Legionella, in fact, is named after an organization, the Legionnaires. So it's actually, the disease is called Legionnaire's disease because there was a Legionnaires convention. So Legionnaires are people who used to serve in the military. And so the name got stuck to the organism. Okay, so just that reminder. Make sure you properly format your organism names, okay? Like I said, you're gonna italicize them, and you're going to make sure you look up the proper scientific names. Sometimes I'll remind you, sometimes I will not, but make sure that you look it up. First time you can use the full name like this, Escherichia coli. After that, feel free to abbreviate it to, abbreviate the genus to E. coli. So make sure you properly format these things in your lab reports. Okay, I have a Kahoot for you. So you can load up the app. If you don't have the app, that's fine. You can use the website, but you can download the app for next time. Let me see here. I got to just pull that up. Okay, here we go. Okay, I'll give you about 20 more seconds to join. Okay, here we go. What does a eukaryote have that a prokaryote does not? So a nice easy one to start off with. A eukaryote is right in the name. It has a true nucleus. Okay, so it looks like we have some points going on. So good job, Gerara. Which of the following are proper ways to name this dinosaur? It should read, is the proper way. There's only one answer. Okay, that one a little more difficult. So the right answer is the one in the blue. So if you take a look at that, T-Rex, that's the genus, and Rex is the species. It's in italics, so that's good. The red one was not in italics. And there's a period after the T to show that it's abbreviated. So what is wrong with the green one? So the green one, you can see the Rex, the R, is a capital letter. It should be a small letter. T-Rex, the yellow orangish one, that's just a kid's book. It's about a dinosaur that likes to drink tea. I thought that one was kind of cute. Hopefully you enjoy that one. Okay, I think I have one more for you. See how the points, Madi is in the lead, good job. In your lab report, species name should be underlined, be italicized, either is okay. Okay, either is okay. Usually typed, the preference is italicized and written by hand, underlined. But either is okay, although I do prefer italicized, but I will accept either. Okay, let's see who wins. Bronze, silver, gold. Wow, out of nowhere. Good job, Shane. Okay, now I just have to figure out how to turn this off. Okay, so let's go back to our PowerPoint. Okay, so a couple other things about organism nomenclature. What do we do about viruses? So I already mentioned that viruses aren't usually considered living, but we do classify them, and they do have a naming system. It's not quite as official, although there is an international committee, believe it or not, but generally this is how viruses are named. Notice they're not italicized. Sometimes you do see them italicized. Usually it's some sort of descriptor and then the word virus, so like rabies virus. Here are some examples. The influenza virus, there's actually an A, B, and a C, so they have a letter written there to distinguish which type they're talking about. Herpes simplex virus 1 causes oral herpes or cold sores, so herpes is the name of the disease, it's right in the name. Epstein-Barr virus, those are a couple of scientists who originally studied the virus. It causes infectious mononucleosis, or mono. Zika virus, so what's Zika? It's a place somewhere in, I can't remember which country. I think it's Sudan, which is in Africa. I can't remember which country, whether that's a village or a river. I can't quite remember Zika. Ebola is a river. Ebola is a river in the Congo, where the disease was first characterized in 1976. What about Norwalk virus? Well, that's named after this lovely town, I think it's Norwalk, Connecticut. And that one actually has been renamed because sometimes people get upset when you name a virus after your hometown. And so now we call it norovirus instead. So you may have heard of norovirus. So what about this new virus that's been circulating, SARS-CoV-2? That actually means SARS coronavirus 2. There was a SARS-1 that was back in 2003, as we call it SARS Classic now. SARS stands for Severe Acute Respiratory Syndrome, so that's the disease. Coronavirus is the family of viruses, so corona does not mean beer. Corona means crown. So the virus itself is kind of spiky and looks a little bit like a crown, apparently. At least that's what the person who named it thought back in the 1960s, the person who discovered the first coronavirus. So usually some sort of descriptor and then the word virus. You can see sometimes that the virus is attached to the word. Sometimes there's a space in between. So it's a little more loose than what you would see with normal organisms. Okay, I'm going to switch gears and I want to talk a little bit about the scientific method. You know, this will overlap with some of the concepts that we're going to cover in the laboratory, right? So what is science? Science is actually the word Latin. It means to know. It doesn't mean we know everything through science. It just means it's a form of gathering and collecting knowledge you know, involves quite a number of things. You're probably familiar with hypothesis-driven science. We talk about the scientific method a lot. But a lot of science is what you would call observation type of science or discovery science. So you can imagine if you're a bird watcher and you are out in the field and maybe you've discovered a new type of bird. You know, let's say it's a green colored raven or something like that. And so now you're documenting, what does it eat? Where does it live? What's habitat does it have? You know, what months is it found in the area? Does it migrate, etc.? You're not doing necessarily any experiments, but you are contributing to the knowledge of mankind. And so this is discovery science. Discovering cell structures, you can see the other example there, gathering genomic databases. There's a lot of examples of that. You're probably familiar with hypothesis-based science. So what is a hypothesis? It's a lot of different kind of definitions for a hypothesis, but usually it's some sort of explanation. You know, sometimes people say an educated guess. I don't necessarily like that. It's not necessarily a guess. It's an explanation, and it's what you think is going on. You know, so sort of like a prediction, but not quite. Pretty close to a prediction, right? And it's something that you know, if you can hypothesis something scientifically, if you can test it, and so you can make an experiment that will actually support that hypothesis, or maybe falsify or cause you to have a kind of modified hypothesis. So here's kind of your basic scientific process. You've probably seen this before in high school. It's the kind of thing that you can start pretty much anywhere along here, but usually people will notice something and then they might have a hypothesis. So let's say, you know, I've discovered a new type of squirrel and it's gray. Normally around here we have those little red squirrels, right? So the question is, you know, I'll observe the new squirrel. Maybe my question is that the same species as the red squirrel? So that would be my hypothesis, right? And so maybe, you know, my experiment will mean catching one or observing that it has the same feeding and habitat habits, maybe observing that it breeds with other red squirrels, those kind of things. Or maybe it doesn't, and you have to come up with a new hypothesis, and you find out that it's not in fact a squirrel at all. You know, it's some sort of oddly colored chipmunk. And then, you know, rinse and repeat is kind of the general scientific process. So here's a little experiment I did when I was your age. I was talking to a friend about these, you know, chocolate cereals, you know, the ones that I didn't get when I was growing up, and him the same. And I'd never tried Count Chocula until I was, I don't know, probably 19 years old. And you know, my friend, he said, you know, Count Chocula is good, but you know, those Reese Puffs are good, but the Count Chocolate is very strong and the Reese Puffs are kind of strong. And I bet you know, if we mix them, we could, you know, make a better product. And so that was our hypothesis. And so we did. And actually it turned out it was great. It was really good. If you're really looking for a good treat, I recommend it. I'm showing this to show you that we do use the scientific method, at least to some degree, you know, on a daily basis. You see something, you think, maybe you test it out, and it's a good thing. Sometimes we have good designed experiments. This is probably a little bit more of a weak one because statistically 2 people, other people may totally disagree. But you know, it's kind of a start. You can see testing hypotheses is not a hard thing to do. Getting good data is where it gets a little bit more difficult, which we're going to cover just a tiny bit in this class and a lot more when you take your statistics classes. So let's talk about good experimental design. So experimental design will include an experimental group. So what do we mean by this? We mean the things that are being tested. So let's say you are testing a new drug and you want to test it on mice. So your group of mice is the experimental group. Let's say you're testing you know, some sort of farming technique and your group of organisms might be potatoes, right? And usually you have a group. You don't want to just test one because there's always weird anomalies. You know, in biology especially, there's lots of weird things going on all the time. You can't just measure one potato and then assume it represents all potatoes out there. You do need to have a large group. You know, the larger the better statistically. What else do we have? We have variables. These are things that are manipulated or changed. And I want to specifically talk about 2 types. We have the independent variable. So sometimes the independent variable, we call this, sometimes people call the manipulated variable. This is what you as the researcher are going to change. So if you're testing, let's say, a new drug on blood pressure in mice, this is what you're doing. You're giving the mice maybe different doses, and that's what you're changing. And then you have the dependent variable. This is the responding or measured variable, and this is what you're detecting. So does the blood pressure in the mice change? Does it go up? Does it go down? This is what you're measuring. Good experiments will have control groups as well. So negative control, you can see negative control groups where no phenomenon is expected. So if you're testing a drug on mice, you're going to have some mice that aren't getting any drug at all, right? You want to just see that, you know, it's not the food you're giving them or the little cage you have in the lab, or maybe, you know, something else going on in the lab or in the field that is affecting the mice and the blood pressure. Positive controls are where, you know, you don't have them in every experiment, but sometimes you do that, and good experiments will have them. And this is where a treatment is already known to produce that effect. So maybe you have another drug that's already on the market, and you're going to test that on the mice to see if your new drug, how it compares, right? You know that the one drug is going to help them with their blood pressure, and you want to see how that compares with your drug. So let's take a look at an example of experimental design. This is Robert Koch. So this is 100 and I was thinking this was 1880s, but basically 150 years ago. And this was around the time where humans were starting to figure out that diseases could be caused by things like bacteria. And he was setting out to prove this experimentally because there was still some debate at the time in terms of what was causing people to get sick. Some people thought it was just bad smells and other things, right? So his hypothesis was that a certain bacillus, so a certain bacterium, is the cause of a disease called anthrax. So what is anthrax? It's something that infects animals, particularly animals that kind of eat things from the ground. So bison are an example of something that have had anthrax in Alberta in recent history. And they dig up these endospores, and it gets up in their nose and in their lungs, and they get a disease in the lungs. So you can imagine any type of livestock that eats grass is susceptible if there's anthrax in the soil. Nowadays we have antibiotics and vaccines, so our livestock aren't getting it that often. But back in the day, this is a very serious issue. There's the bacterium, and so Koch was seeing this bacterium, and he was seeing a lot of the sick animals or dead animals. And so he asked that question: how can I test this hypothesis? So I'll show you his experiment. It's actually quite simple. But this kind of thing had never been done before at the time. So you can see here we already have 2 groups, right? We've got a healthy organism and we've got a diseased organism, right? And so here he is, and he's finding this bacillus, this anthrax bacterium in the blood of the sick animals. So he takes that organism, he grows it up in a petri dish. So by the way, like petri dishes weren't even a thing at the time. Julius Petrie was actually one of the lab assistants who actually invented the Petri dish. So he had a lot of hard work ahead of him to try to prove this. Next step was pretty simple. You inject it into a healthy animal and you see does it cause a disease. And so, you know, this idea that the diseased animals are the only things that have that particular bacterium, and he was able to prove this to the world, right? That anthrax, the bacillus, causes the disease anthrax. So let's just pick this apart a little bit. This experiment, right? So we've already kind of, you know, you can sort of see the hypothesis here, right? The hypothesis was that that bacillus anthracis causes anthrax. All right, so that part is probably pretty obvious. That was the hypothesis. It's a pretty straightforward hypothesis. So what are you thinking are the experimental group? Well, in this case, the animals. Initially, he tested it on mice. Eventually, he did test it on livestock. Mice are a little cheaper than livestock, so you're not going to go right to the livestock. You can do something cheap like mice, and they're basically vermin, so nobody cares about them. So what is the independent variable? So you got to go back and think what that means, independent. Independent means the variable that is not going to change with the experiment. So that's the variable that the researcher is controlling. So in this case here, this would be the presence or absence of the bacterium. And the dependent variable would be the presence or absence of the disease. So pretty straightforward. In terms of controls, I don't know if we actually know what his controls were historically. Presumably, he did have a negative control. That means mice that were not getting injected, but were living in the lab, being fed the same food, being treated in the same manner. Whether you had any positive controls, no, I don't think so. We didn't have anything else that causes anthrax, so you can't have a positive control for that. You'd, of course, control a lot of the environment. Like I said, you're feeding all the mice the same food. You're probably using all the same strain of mice, etc., and things like that. Okay, so I know we're running out of time. Let me just see. Oh, I do have this written on the slide. There we go. I didn't realize I had done that. So there we go. I basically wrote the exact same things. Okay, so I know we have a couple minutes left. I just want to wrap up with this, some thoughts. Can we always use the scientific process? So what about these kind of questions, right? You know, let's look at that one about the dinosaurs. Unfortunately, we can't just make a planet and put a bunch of dinosaurs on it and see what makes them go extinct. We can't do the experiment. But we can do experiments around hypotheses. So you may know that the big hypothesis around the dinosaurs is that an asteroid struck. So maybe that's your hypothesis. Then you can go out and look for evidence. And that's hypothesis-based science. So you can look for, let's say, a meteor strike crater. Let's say elements that are in meteors found around the Earth and things like that. You're not going to be able to prove it, but you might find evidence to support it. Some questions are a little harder. Science can't answer everything. Of course, we all know the answer to that first question there. Ha ha ha. Just kidding. You know, how do you even measure intelligence? It's a huge debate, even amongst psychologists. What is love? Is there a God? You know, I'm pretty sure my wife loves me, but I don't think I can do an experiment to do that, or that might be unethical. So just something to think about. I do have another experiment here. I'll maybe cover it next day. I just want to show you that at the end of my notes, I have kind of some concepts to think about, some terminology. The terminology is super important for your test. It's very important that you know these words. You're going to be asked about them directly. There will be definition type questions on the test that I will take from these word lists. And it's important you know what these words mean. If you're answering a multiple choice question and you can't remember what the word taxonomy means, you're going to be in trouble. So at the end of every set of notes, take a look at these words, look them up in your textbook, Google them, figure out what they mean. Very important part of your studying. I also have some study questions. These study questions are not exam questions. They are study questions. What does that mean? It means these ones are kind of like bigger picture kind of questions, right? You know, what is science? I'm not going to ask you what is science on the test. It's going to be a big long essay, and you're just not going to have time for that. But I'm going to look at these questions, and I'm going to use them to inspire my test question. So you can see question number 2, what is a cell? What are some characteristics of a cell? You know, probably won't ask you that. That's a little vague. But maybe there will be a question asking you to compare eukaryotes and prokaryotic cells, for example, right? Or maybe, you know, something even more specific than that. So go through these study questions, you know, when we go through every topic, and make sure that you understand what's being asked of you. And this is the part for discussion, and this is where you can, you know, contact me during office hours and find out, hey, what do you mean by number 6? I have no idea. When did we talk about that? And I can try to help you out. So I think that's the end of topic 1. If there are no questions, I'm going to finish there shortly. Just a reminder, if you've not done so already, please do your plagiarism certificate for next week. And we will talk about the lab. Once we get into labs, I'll probably spend a little bit at the beginning of every lecture kind of talking a little bit about lab report writing and things like that. If I gave you all the tips at once, you know, your brain is just going to be overloaded. So just little tips and little examples of things that have been done by previous students, you know, what looks good and what doesn't look good and things like that. Yeah, so I think I see a question there. Is this slideshow on Moodle? Yes, it is. I will be trying to post them before I give the lectures if I can. It might only be 5 minutes before, but I will post them on Moodle. Plus the meetings are supposed to be recorded, so unless that fails, I will also post them on I have a YouTube playlist that's linked in Moodle. Anyway, hope you have a great weekend. I already need, you know, I've only been at work for a couple of weeks now, and I certainly need that long weekend. So hope you can enjoy yourself and get ready for the semester. It's going to be busy, particularly if you're taking 5 courses. It's going to be quite a busy semester for you. Anyway, have a good weekend and take care. I'm going to turn this off now. Oh, I saw one more question. Do we have any homework? Just do your plagiarism certificate. That's what I want you to do, and look at that email about the labs. Anyway, have a wonderful weekend. Take care and enjoy the little bit of sun we're getting today.